Okay. I'm gonna move the captions over. So, supposed to be. Okay. All right. Good day, George.
Let me just check really quick. This is, how do I progress slides?
It should sometimes.
There
Okay,
you
yeah, it got mad because it moved the captions. That's all.
go. Okay, no worries. All right. Hi, everyone. My name is George Zaragoza. I'm a third-year PhD student at the University of Central Florida. And today I'm going to be talking to you a little bit about the work I'm doing currently with Florida key deer. All right, so key deer are subspecies of white-tailed deer, so Odocoileus virgianus clavium. They're currently a ESA listed endangered species. They faced a lot of high hunting pressure about in the 1950s, dwindled down to a population size of about 25 individuals. But since then, their population has increased in number. One of the most obvious traits about them is their small size. So as full-grown adults, key deer reach about 25 to 30 inches. So this is relatively similar to the size of something like a golden retriever. And adults reach anywhere from 70 to 80 pounds. So really, really small deer, much smaller than what you'd see or expect from the white-tailed deer up north. All right, so key deer encompass a range that's very, very small. So you have the state of Florida, and then down just past the south tip of Florida, you have the Florida Keys. And in the Florida Keys, the key deer are isolated to these about 20 islands in the Keys surrounding Big Pine Key, which is this key that's starred. And so the key deer will actually swim from key to key, and then swim as far down south as Key West, which is the furthest south island, and then as far up north to some of these keys that are a little bit closer to the mainland. But about 65 to 70% of their population is actually located on Big Pine Key. All right, so one of the interesting things about key deer is that over the last 5 to 7 decades, they've actually experienced a lot of urbanization in the Keys. So the Keys have become a really popular tourist destination. They faced a lot of development in the last few years, or the last few decades. And during that same time, as development has increased, that population size has also increased. Partly this is due to the relaxation of predators like coyotes and bobcats. As people moved in, as tourists move in, people wanted those bobcats and other mesopredators out of the area, so those are no longer really around. So there's that decreased pressure, but interestingly, there's more and more of a dependence on urban areas. So in general, key deer you see spending more time in these urban areas or in places closer to the center of towns. And so a paper in 2007, Harvison et al. Basically compared historic and current populations of key deer. And what he saw was, looking at historic populations of key deer, their actual urban use, as far as the proportion of urban use during the day, was pretty low in general, but their survival rate was about 0.9% from year to year. But then looking at current key deer, there's now a more direct relationship between the survival rate and urban use. So the more time the key deer are spending in these populated areas, the higher rate of survival they have year to year. Part of this is because people are now developing relationships with this key deer, actively feeding and watering the key deer, and human watering bowls are pretty much the only source of fresh water the key deer have on the islands. Thinking about genetics, so Ellsworth et al. In 1994 looked at different populations of white-tailed deer in the southeastern United States, and basically compared all these populations and genetic relatedness using a few genetic markers. And what he saw was that the Florida key deer was the most divergent group of all these southeastern populations. And they also had these unique heritable variants that were only present in the key deer population. In 2017, Vicky Villanova et al., she did this kind of 2 studies in parallel, one looking at the demography of key deer using marker recapture studies to estimate population size. And she also looked at the genetic variation, looking at effective population size in the key deer. So what she saw was that the number of individuals based on those marker recapture studies was about 1,000. However, when looking at the genetic variation and thinking in terms of this effective population size, this is equivalent to the population genetic variability that be present only in 11 individuals. And for those of you familiar with genetics, this is in a situation where you reach Hardy-Weinberg equilibrium. So that in a nutshell basically tells us that this population of key deer is very, very inbred and likely in the past has experienced a lot of different bottleneck effects, especially due to that high hunting pressure in the 1950s. All right, so my research questions, Previously, those studies have focused on these small sets of genetic markers, you're looking at about a dozen or so places in a genome. For my study, I'm focusing on a whole genome, so looking at the entire genetic sequence of key deer. And so there's three questions that I'm focusing on. One is, are there genomic regions unique to key deer versus white-tailed deer? So thinking more broadly in terms of those unique those unique genetic variants that were found in the previous studies, but now looking at entire genome and encompassing as like entire chromosomes as we can. We're also interested in thinking about which regions are detrimental and which regions are adaptive. So when looking at an inbred population like this, often, especially if it's associated with proximity to humans, you see more of a tolerance for these detrimental mutations, or they're present in higher proportions compared to what you would see in a quote unquote more wild species. And then third, are there patterns of adaptation similar to urban adapted or domestic species? So here there's three different things that we kind of want to look at. There's different pathways that have been hypothesized to be associated with urban adaptation to domestication. Examples of these are like the neural crest cell hypothesis, looking at like development early in the embryo, thyroid hormone production, as well as stress hormone productions like cortisol. So there's a few well-known genes or markers that we can look at that then compare our key deer data to other species that might be domestic or urban adapted. All right, so for our research method, we used 11 key deer that were originally sampled from Villanova et al. In 2010 to 2013. And these were mostly found in Big Pine Key. So this larger key down in the south, in that big area that they kind of spanned across. We took that tissue, which was, they were key deer that were darted, that skin tissue was then digested, and then we extracted the DNA from that tissue. After extracting the DNA, we sent that DNA off to sequencing. We used a third-party company to do this, Novogene, and used Illumina NovaSeq, which is a form of shotgun sequencing. And so for those of you unfamiliar with like kind of these sequencing methods, so just in a nutshell, shotgun sequencing is a really popular method because it generates a lot of high-quality data. And so what we do is we start with a sample of DNA from a key deer like so. And in that sample, you have many, many fragments of DNA. So various size fragments, some are smaller, some are larger, but there's a lot of DNA in that sample. As you put that DNA through a sequencer, the sequencer then translates those bases into these ACTGs that we're familiar with that become the actual genetic data that we use. And so you do this for all of your fragments in that sample. And then by comparing it to a reference genome, you can then map those fragments to a reference genome. You're basically matching the patterns of those fragments to similar patterns in that reference genome. So over time, those patterns match up. And then as you stitch them all together, there's a little bit of overlap. So you eventually get something that looks like an entire genome. So this is your new novel genome that you then created from your sequencing data. However, there's often variants present, so it's never exactly the same. And these variants that occur, if they occur in population, across the population, or in individuals compared to a reference, are what we would call single nucleotide polymorphisms, or SNPs for short. All right, so after sequencing, we then downloaded that data and put it through quality control using a software called FastP, basically making sure the quality of the sequencing was high enough to allow us to use that data as our actual analysis. It turned out like 99% plus of that data was actually really high quality, so we were really happy with those sequencing results. We then moved that over to our mapping step. So mapping, like I mentioned previously, we're putting those fragments and comparing them to a reference genome using a software called BWA. And then we ended up processing and filtering those using a software called SAMTools, which basically is just cleaning up, removing duplicates and redundancies in the data. And then finally, we identified variants using a software called BCF tools. So this is identifying those actual SNPs that are present in the data. All right, so looking at our sequencing data, the quality that we're looking for in general is a quality score of 30 or above, which tells us that our data is good enough to use for these types of analyses. And overall, what we saw as the sequencing continued to progress, so the longer the cycle, the more cases are sequenced. And over time, what you expect is a general degradation, but for the most part, all of our samples were pretty high quality above this 30 marker that we had set. Coverage depth is something related to the overlap of the sequencing. So again, compared to that reference genome, if you have many, many fragments, occasionally there's overlap. The more overlap you have, the more confident you are that that specific position in a genome is actually the correct assigned base. And looking at the data that we actually produced, what we saw when comparing key deer to white-tailed deer was pretty similar. So we had a genome size of about 2.5 billion compared to the 2.4 billion of the white-tailed deer. But this data hasn't been processed through some of these steps that clean up anything associated with potential contaminants or bacteria or things like mitochondrial DNA. So the data is high quality, but there's other stuff that might be floating in there, which I expect would drop that number a little bit closer to the white-tailed deer. GC content, just the proportion of our GC bases to our GCATs was about similar between white-tailed deer and key deer. Using BCF tools, the software, we identified 40,000 single nucleotide polymorphisms, which is great because when you think about those earlier studies I mentioned, they were only looking at about a dozen markers, a dozen of these SNPs. So we have thousands of times more data than they did. And now we can really address those really deep population genetic and adaptive questions. All right, so continuing work in progress. So I really tried hard to get this today or to get this produced today, but I just couldn't. I probably still need another week to actually produce this, but we use a software called SNP-F, which actually annotates these SNPs and then predicts their function based on the gene in which they occur. And so basically you would get a nice list of different genes and what variants were associated with those genes and what those genes could be potentially doing in the behavior and the physiology of the animal. This is just an example from a study that's done in warblers in our lab. But what we really want to do is compare functional differences between key deer to white-tailed deer and other domestic or urban adapted species. So when you think in terms of missense, nonsense, and silent mutations, your missense mutations are basically when there's a change due to a SNP from one amino acid to another. Your nonsense is when there's a change from an amino acid to something like a stop codon. So you essentially break your gene. And then your silent mutations are when there's no change. So usually you have a really high proportion of the silent mutations. What we expect in some of these more domestic species or these potentially even urban adapted species is a higher tolerance for these missense or these nonsense mutations. And so these proportions could be potentially higher, and that's kind of one of our hypotheses with these key deer. We're also going to look at historical samples from the Florida Museum of Natural History. So these are samples that were collected prior to development in the 1950s that we want to take this bone from the key deers from then, take them to an ancient DNA lab and actually extract that DNA and get it compared to our current contemporary samples. During November of last year, there was a workshop by the Society of Molecular Biology and Evolution, SMBE. They had a workshop in urban omics, and basically it was like an unconference where you had a bunch of different working groups that were focusing on brainstorming different perspective papers. And one of them that I was co-leading was looking at this domestication continuum between domestic and quote unquote wild species, and really asking, does this actually exist? Is there some continuum between domestic species and wild species? Do we see the abundance of certain traits associated with these domestic species that might not be present in wild species? And how does that compare to things like commensalism, pseudo-domestication, and urban adaptation? So really kind of up in the air, just going to be looking at a bunch of literature and trying to synthesize what this looks like, and if there's actually potential evidence of this, or if there's kind of situational case-by-case basis for this. All right, so some additional considerations. Yes, there's more development in the keys, and there is more deer associated with the development, especially looking at the last 7 decades or so. But the number of individuals doesn't mean that that population is actually genetically stable. So that population, like I mentioned before, it's equivalent to the genetic variation found in a population of 11 individuals that were breeding, that were admixed and breeding normally. There's also, because of the high inbreeding, a high disease risk and high risk of bottleneck effects in that population. So if a disease actually were to come through in the key deer population, it very easily could wipe out the majority of that population. Also, it's important to understand the history of genetic variation when looking at a population and thinking about conservation and preserving a species, because we want to know what base to go back to. If we think of protecting the key deer, yes, today they might look a certain way, but maybe 50 years back they might have not, and that genetic variation that we see in the genome might look different compared to what we see today. And so we want to preserve that and understand how that looks like compared to today in order to really think as best as we can about what it means to preserve a species or preserve the genetic variation in the species. We also want to disentangle the effects of inbreeding and adaptation. So this is something that can be done using a coalescent analysis, which you're basically looking at a family trees of these genetic variants and looking back through time how they're related and kind of condensed back to their ancestors. And using the historical samples, we plan to do this, but then we have a ballpark basis to confirm with these coalescent analyses. We also want to look at demography compared to white-tailed deer. This is primarily done by Dr. Aaron Schaefer's lab at Trent University. So I extracted a bunch of key deer for them, sent them over, and they're currently doing a phylogenomic analysis across North America of white-tailed deer and these island populations and other isolated populations that exist. But thinking more in terms of the demography, how these populations might have evolved over time, and if there's similarities between these more divergent populations. With this data and the data that we have about 25 individuals total that we can look at, including 2 historical samples, and that we can use to address these demographic, more population genetic questions or questions about adaptation, which we plan to do in our lab. All right, with that, that's all I had. Just want to say thanks. So, so special thanks to my lab, Dr. Vitak is my advisor. Hoffman Lab was the lab where the samples came from. Florida Museum of Natural History has been really, really great in communication with us and having us see what samples they actually have. And then SMB workshop was really, really great for getting a bunch of people to collaborate and really think and work on these topics. If you're interested in working in this budomestication wild group, feel free to email me. We're still putting organizing things and code leading kind of the beginnings of this as we start to frame this perspective paper. But thank you.
Time for questions if anyone has a question for George.
Excuse me.
Yeah.
So this comes from just me being kind of ignorant about this.
Let me do this to make sure. Okay, you should be good. All right, thank you. All right, hi everyone. I'm Jess Proctor. I'm a master's student at SUNY ESF out of Syracuse, New York. I'm co-advised by Dr. Brad Constantino, who will be presenting in 2 sessions. So if you didn't get enough of squirrels from me, stay tuned for more. And then Dr. James Gibbs as well. So I am the second of this squirrel trilogy that Adam alluded to this morning. So there's lots of squirrel talk today. We're part of a bigger project. But my major interest is looking at crypsis and how that can be explained across urban to rural climate or urban to rural gradients. So getting started, urbanization and urban areas are the most rapidly expanding systems in the world. We know this, and we know that they have a lot of effects on the landscape, right? Whether that be habitat fragmentation, pollutants, both chemical and light. And we also know that these pressures can affect wildlife. Obviously, we're having a whole conference about urban wildlife. So specifically, I'm interested in animal color and how urbanization may be impacting animal coloration. So taking a step back, animal color is important and plays a lot of important roles in nature, including things like sexual selection, thermal regulation, and then my area of interest, camouflage, right? So how well individuals are blending in with their habitat. And there's plenty of great examples of how urbanization can impact animal color. First, we have the example of the great tip on the far side there, and can you see my cursor? No, I'll walk over. So in polluted areas, they'll lose this yellowish coloration and have this more urban graying effect. And then if we have arguably the most famous example, the peppered moth of coloration and urbanization. So as we have more polluted environments, this black morph is dominant. And this brings in this idea of this background matching hypothesis. So perhaps it's advantageous to blend in well with your background, more so than your neighbor who's this bright white moth on a dark tree. It's going to get picked off by a predator quicker. So my research is looking at that idea of this background matching hypothesis, and my study subject is the eastern gray squirrel. So there's very few photos of a black and gray squirrel in the same picture. So through our presentations, you'll see repeats of sharing pictures. So anyway, historically, this melanic morph, this black morph of the squirrel, was dominant across the landscape. But now what we're seeing is this climb in melanism. So as we approach city centers, there's more likely probability of having a melanistic morph. And that includes my study site, which is in Syracuse, New York, and that's where my presentation will focus. So what's going on here? And my major question is, can we use crypsis and this background matching idea to help explain these trends across the urbanization gradient? So in order to address that question, we really need to step back and understand what that environmental gradient is and the implications that these different habitats have on squirrels. So starting with the most rural of habitats, we have old growth forests, which are relatively rare in the landscape these days. And what we have mostly in the Northeast is what we call secondary forests. So these are younger forests, more open canopies, shorter lived species, and more light. We also know that predators are a limiting factor of squirrels in these forested habitats. So perhaps when these darker morphs were more widespread, it was in an old growth forest where they could blend in better. And now we have these more open, lighter environments where they're standing out and being picked off by predators, making them less likely to survive in these more rural environments. And then on the flip side of that gradient, we have these heavily urbanized areas. So downtown city centers and where there's lots of development and high density of roads. And we know in these environments that predation is likely relaxed and that vehicle collisions are a major threat to squirrels. So perhaps in these environments, it makes sense to stand out, right? It doesn't matter what color you are, if predators aren't going to pick you off as aggressively as they will in the forest. And by standing out, you're less likely to get hit by a car. So these are working hypotheses. And then in that gradient, we have these urban green spaces. There's something somewhere in between downtown and highly developed and these truly rural environments. So I worked across these habitats, across this gradient in and around Syracuse, New York. At 110 different locations, I took thousands of photos of squirrels and all sorts of different positions. So I took pictures of squirrels climbing on trees, running on the ground, and then running at some elevated structure, whether that be on a branch or in a yard, like someone's roof or something. And then I also took pictures both at the ground level, so like human perception, and 20 feet above on a tripod aerially looking down to mimic like avian predators. So in all these photos, I standardized for lighting and had like standardized camera. And for every picture of a gray squirrel, I replicated it with a melonic in the exact same position under the same conditions. Additionally, because why not take more pictures, right? 1,000 wasn't enough. I took photos in different seasons. So leaf on, and then again, once the leaves fell off the trees. So the question I usually get at this point is, Jess, how did you get all of these squirrels to so perfectly pose for you? And I brought my own. So I had a fleet of taxidermy squirrels. I'm a weird wildlife scientist who actually just didn't interact with real wildlife. I guess they were formerly real, but so I would take these fleet of taxidermy squirrels matched for size and bring them into the forest. Take the picture of the black squirrel and then switch it out for the gray. These guys are pretty realistic. I got stopped by a forest ranger once who thought I was out poaching squirrels illegally because he looked into like my hatchback and just saw like 12 squirrels laying across it. So fun fact, if you're going to like hang taxidermy in the forest and take pictures of it, wear a reflective vest because people just think you're a professional and they don't think you're like performing witchcraft like they do when you're not in neon. So some examples of what those look like. This is a picture of a gray squirrel in an old growth habitat. And then there's the black squirrel counterpart. So for every picture, we have that pair. And here's an example of an aerial shot. There is a gray squirrel in this photo, I promise. It's a little bit easier with the circles. So comparing the gray is almost invisible on that aerial concrete shot. And then importantly, also looking at roads. So pair between a black and a gray on a road. So I use these images in two different ways to measure crypsis and how well these squirrels are standing out. So the first was, I developed a game, it's called Squirrel Spotter. If you haven't played it before or never heard of it, yeah, we know we have some Squirrel Spotter fans here. So that's a QR code. There's a bigger one on the last slide that works. You can scan it, it will pull it up on your phone, you can play it. Sometimes it's better to play it on the computer. It's sometimes a little small and hard to find the squirrels on your phone, but there she is. So basically users see a random set of 30 images, and then the game will measure detection time, how long it's taking them to detect the squirrel. So we have these paired black and gray, and we can use that to measure the difference in detection across these habitats. So what does that look like? It is important to note, let me just go back real quick to this. When I was taking pictures in the roads, I only had one perspective. I didn't take photos from 20 feet above because I didn't want to be standing in the road with a tripod. And only one position running on the ground. So in analyses, I did pull out roads and analyze the other environments separately, and then did a solo road analysis. So I broke this down because this is a lot to digest, but we're really interested in that morph environment interaction, and then how that interaction responds to some of these other variables. So we found a significance in the interaction of morph, environment, and position. So I have these graphs broken down by detection times based off of positions. So starting with climbing, just to orient you to this graph, the y-axis is a delta. So it's the difference, the detection time difference between finding the melonic squirrel and the gray squirrels in a habitat. So that dotted line above it means they would find the gray squirrel faster. Below it, they found the melonic faster than gray. And what we see here is some variation across environments that's leading to that significance, but still that trend that everything, all the black squirrels are found faster than gray. Again, we see that in ground, and again on branch. There's a slight difference with those urban yards. It's not actually a significant difference, but still very, very small. And the trend is very present of black found faster than gray, regardless of habitat. So this was based off of like 2,000 unique gameplay users. And then looking at roads, because there wasn't all those additional perspectives, we just looked at morph and season. And again, same trend, black found faster than gray across seasons. There's some variation between leaf on, leaf off, but general black faster than gray. So this leads to some interesting implications. I'll circle back to them at the end, but for sake of time, the other quantitative measure was I took quantitative estimates of background matching, which is a mouthful, but basically I used machine learning to train a computer to tell me if a pixel in the image was a closer match to a black squirrel or a gray squirrel. So what that looks like, trained to the black squirrel picture, trained to the gray, and then I would classify it over the entire image. And it gives an output like this. So the red is a closer match to gray, and green is a closer match to the melonic. So we looked at the total background, so the entire picture, the total complexity of the scene, and then also that immediate background of like the tree the squirrel is climbing on or whatever's immediately behind it. So it's important to note, I'm still in the process of this machine learning. It takes a hot second to go through thousands of photos to train this. So this is a working data set. I have about 6 environments or 6 scenes per environment, and there's 20 total. So take all these results with a grain of salt. So first, looking again, pulling off the roads, just looking at the other environments. This is just looking at proportion black match. So 0 is there's no match. It's a closer match to gray across the scene, and 1 would be a 100% closer match to black squirrel. And what we're seeing here is across environments that there's a low percent match to the black squirrels. And then switching over to roads, again, looking at that seasonal leaf on, leaf off, still a lower percentage match to black, more close match to gray. So going back, I skipped over this, but looking at this quantitative estimate, we found when looking at these other environments, excluding roads, the immediate background, there was significance, but when you zoomed, or the entire background, sorry, but when you zoom into the immediate background, we found no significance across environments. And we see the opposite in roads, right? So when we look at that entire complex scene of the road and everything surrounding, there's no significance between seasons. But when you zoom in and you're just looking at the immediate background of the morph, that's where we start seeing significant results. So squirrel background matching directly on the roads versus the entire complex road environment. So that was kind of a mouthful. I'll take questions so I can explain that better if things were unclear. But just to summarize, we're finding melonic morphs are found faster and are exhibiting less background matching than gray morphs across all environments. So what does that mean, right? We talked about this background matching hypothesis, where it might be more beneficial to match your environment. But across everything, these guys are standing out more, right? So what does that mean? We know squirrels are in urban, these melanic squirrels are in urban areas, but absent in forests. Maybe predation's relaxed in the cities. Maybe it doesn't matter what color you are because the predators aren't attacking squirrels aggressively. But versus in the forest, when you're standing out and everyone's detecting you faster than the grays, then you're more likely to get picked off by predators, and that's excluding these squirrels from rural areas. And then also maybe there's advantage to actually being more visible in these urban environments so that cars have more time to avoid you on the roads, and that's the data that Adam is working with. So next steps, first, obviously combining this data with the roadkill analyses that we talked about this morning. And then also figuring out predation rates across these urban to rural environments. We need to understand if predators truly are relaxed in urban areas versus in the forests. So that being said, we've been trying to figure out how to make models that predators will actually attack. So if anyone knows someone who's really good at taxidermy, we need more taxidermy squirrels, so send them our way. Cool. All right, so thank you so much. Again, here it is, really big. If you haven't played Squirrel Spotter and you're interested, now is your chance. Thank you to my co-advisors, the squirrels, of course, all the homeowners who let me take pictures of squirrels in their backyard, and all of the citizen scientists, and hopefully any of you who are going to play the game as well. All right, thank you.
Okay, and we've got time for questions if anyone has a question for Jess.
Yes.
I think your analysis of sort of the different scales is interesting, like the background matching of like the total.
Cool. All right. Well, thank you very much for being here. I know it's the last kind of day and we're all getting a little tired and stuff, but if you were at my previous talk, a lot of this is going to be relatively similar, looking at a different question, but using a lot of the same data and really kind of going within the same realm. I'm really in a dial activity kick right now, so I'm focusing on that quite a bit. But for this particular talk, I'm going to hone in on kind of something different than what a lot of my other research has been, which is like interspecific interactions. For this particular project, I'm going to be looking at intraspecific interactions and the variation within species that is often overlooked when we talk about how a species reacts to urbanization. And so we're going to be focusing on mule deer here. I also realized I don't put my email on like any of the presentations I do, and so I quickly added it right there. So if you want to reach out to me about any of this, that's the best spot. There won't be another email throughout the rest of the presentation. Okay, then let's make sure. Okay, cool. So this is my study area. I am in Utah and I'm working in Salt Lake City. And Salt Lake City is a really unique landscape in that it's kind of a massive juxtaposition between undeveloped rural land and urban land. I would say highly developed, but being, having people from the Eastern United States here, this is definitely not highly urban in any way, shape, or form. But it's a metropolitan area and there's a lot of urban development in this spot. And it is nestled right up against a lot of highly undeveloped areas. So it's this really stark difference from these two dichotomies. And so it allows for direct comparisons between the effects of urbanization in a more rural landscape, really right at a really stark interface between the two. And so a lot of what I do looks at questions that are related to what is going on as animals go across that gradient, as they start entering into the city center versus going out into the Wasatch Mountains, which is that surrounding mountain range right there. And, this is preaching to the choir again for everyone that is at this conference, but occupying urban environments is a biological trade-off in a lot of ways for a lot of different species. I'm only going to focus on one particular one for this talk, and that is because this talk's all about mule deer. And so one of the big things for mule deer, one of these big trade-offs that they have to deal with when they occupy urban environments is they do have this decrease in predation. So they're no longer or potentially released from the risk of predation from their top predators, in this case, something like a mountain lion. But on the flip side of that, they now have to deal with human-caused mortality, or at least the threats of human-caused mortality in a lot of different ways. And so being able to kind of navigate through that trade-off is a really important thing that mule deer have to be able to do in order to come into Salt Lake City. And coming into Salt Lake City may be a benefit when you consider the landscape being so seasonal, whereas in the urban area, these resources are available at a much more consistent basis. And so one of the ways that mule deer may do this is they may alter their temporal activity behavior. And again, if you've been to my previous talk, I'm sorry, this is all very, very similar to what you saw before, but I have different pictures, so I thought that might be a little bit better. But really, when we look at temporal activity behavior, it's a very important component of any species ecological niche. Just like when we look at how species occupy space, if we don't consider how they're also using time, we're missing half of the picture of how they interact with their environment. And because of this, a lot of species that may or may not be able to occupy urban environments, they do so with their ability to change when they're active. And so it is important on things like community structure within urban areas. It's a very important component that tends to be overlooked. And one of the biggest things for this particular study in question is that not all species, at least in Salt Lake City, are adapting to urbanization in the exact same way. So this was a previous investigation where we simply just said, okay, how are species adapting? And is this leading to interspecific differences between them? And if so, is that changing the interactions between species? And so what you're looking at is a bunch of different graphs that show just temporal activity distribution. So on the x-axis, you see time. On the y-axis, you're technically looking at probability density, but all you need to know is that as the curve goes up, the activity of that species increases. And so we have a curve for Salt Lake City, we have a curve for the Wasatch, which is our more rural area. And then you have the overlap underneath. And the biggest thing I want to draw your attention to on this graph is that every single species is adapting to that Salt Lake City study area in a slightly different way. So they're occupying different parts of the day in order to adapt to those areas. And it tends to follow how they're either trying to avoid humans or each other. And so just to give you a few examples, we have our already nocturnal species, and they didn't alter their activity much at all. They already were fairly adapted to avoiding humans, and we see relatively little adaptation in that case. But when we look at specifically, let's focus on coyote and mule deer, we see that coyote became almost strictly nocturnal as they came into Salt Lake City, which makes sense. At least in Utah, there's still a bounty on coyotes. And so it's basically a standing kill order. If you see a coyote, shoot a coyote, and everybody in Utah has guns. So it makes sense that they're going to avoid coyotes or avoid humans as much as possible. So they're really active at the kind of dead of night. But what we see with mule deer is they actually increase their diurnal activity as they went into Salt Lake City. And it made this really weird trimodal curve, which if you know the physiology of an ungulate, doesn't really make a ton of sense because they need that time to rest in between periods of activity to actually digest their food. So what this was suggesting to us is that what we were picking up in Salt Lake wasn't necessarily the behavioral change of just one species in general. It was the behavioral change of two different populations within that particular species. And what I mean is a lone adult population and a breeding population. And so there may be differences between that, because then when we looked more closely into the mule deer curve, we saw that what was driving that increase in diurnal activity was actually sites with increases in coyote activity. So it got us really thinking that there might be something going on here. And then in 2019, this paper came out for white-tailed deer, and this is just basically temporal activity overlap. So this is looking at the overlap with coyote and for white-tailed deer. And it's just looking across different life history stages. So we're looking at lone adult bucks, lone adult does, and then nursery groups. So does that are actively raising young, and then the overlap with the most common predator in the eastern United States for them, coyote. And we see it decreases as we go into the nursery group level. So something's going on there with this intraspecific variation. And so this posed the question for us, are fawning deer, so mule deer accompanied by young, are they reacting to urbanization differently than their lone adult counterparts? And if they are, is any of that being driven by urban predation from coyotes? in that particular area. So this was really the focus of this particular project. And to get specific with the questions here, how does urbanization alter temporal activity patterns in lone adult versus fawning mule deer? And if intraspecific differences occur, what are the factors that are potentially driving those differences? Our major working hypothesis is that based on that previous research that I showed, we're most likely going to see a difference in urbanization response between the breeding and the non-breeding individuals. And we were predicting that the fawning individuals were going to respond to a larger extent and become more diurnal. And some of this may be able to be explained by avoidance of coyotes. And so that was really kind of our working theory. Okay, so for results, these graphs get kind of intense, and I should have just kind of walked through one by one, but I'll still kind of walk through them. So this is just for urbanization. This is lone adult mule deer activity. And of course, every time I put something up here, something isn't shown. In this case, it's the overlap, which would be a light green, but it's okay. At least the curves are shown. So what you're looking at here is those same type of temporal activity curves. So you have time on the x-axis, activity distribution on the y-axis. Then you have the two different curves for the two different study areas. Our rural study area is the Wasatch Mountains and surrounding national forest. Our urban study area is the Salt Lake City metropolitan area. And so what you're seeing is the overlap between those for lone adults. And although the difference is statistically significant, there's also over 25,000 mule deer detections in this data set. So just about any difference at all was going to come out. So what I really want to draw your attention to is the magnitude of that difference, which is relatively small. So the overlap coefficient on the top there ranges from 0 to 1, and 0.92, that's a pretty strong overlap between these two populations. And we look at total activity, we see that that's the total amount of time spent active. The urban population's active more often than the rural population. And then for core activity, that is the smallest amount of time where a majority of detections are found for both of those populations. They remain relatively stable across those differences. If you want to talk about why the urban activity is higher in lone adults, I'm happy to do that because it'll make more sense when we look at the fawning activity. But now when we look at fawning mule deer, it gets a little bit more interesting. So we're looking at fawning mule deer in this case and the differences between their activity curves. And again, we see a pretty stark change in their distribution of activity. And we see that the magnitude of that change is also much larger. And so then when we look at the right side of this curve, we see that the urban deer are actually now active less often than their rural counterparts, as opposed to previously with the lone adult deer. And they've drastically shifted the times of day that they're most active, their core activity there. They're active a lot more in the early morning to kind of late morning, and then they kind of halt that activity towards the later evening. So then when we compare those two together, so now we're looking at the comparisons of lone adults versus fawning. The top is rural, the bottom is urban. We start to see that it's urbanization that may be driving some of these changes. It's the moving into these urban environments, because on the rural side, they're relatively similar. Their distributions are similar. They have relatively the same amount of time spent active. Their core activity is relatively similar, but it really starts to change when we look at the urban activity. And in particular, I find this side of the graph relatively interesting because I think it's kind of intriguing that total activity within fawning deer is different than lone adult deer, and that core activity has shifted substantially. So then the last thing was, okay, is predation potentially driving any of this that we're seeing? And so these are the predictors for the lone adult activity during different times of the day. This is a multinomial Bayesian regression, it's really just a fancy way of saying, okay, let's see what is affecting these different time periods of activity. And for the most part, we see what we'd expect to see for any type of large mammal. A lot of the environmental predictors that are associated with human density or impervious surface cover coming out as negative. NDVI is kind of curious in this case. So if you look at NDVI within Salt Lake City and really within Utah, the spots that kind of shoot up as high levels of NDVI are actually within the city instead of outside of the city, just because we are in the desert. And so it's a kind of an interesting way to look at that. But with the fawning activity predictors, so everything highlighted in green had the same magnitude and the same direction of change as the lone adult counterparts. The lone one that was included in the top model for fawning dial activity and not included in the lone adult was coyote rate. And so now these are the 95% confidence intervals. They do overlap. We were looking at more of a one-sided test though in this particular case because we had a decent idea from previous research. And so this was still deemed statistically significant if we look at that kind of one-sided activity. But we see kind of what we might have been expecting to see is that as coyote rate increased for fawning dial activity in every time period except the nighttime, we saw a decrease in fawning mule deer activity. And so to kind of wrap this all up, we had our general hypothesis. And in general, that hypothesis was supported. We did see that fawning mule deer did tend to change their dial activity patterns in response to urbanization more so than their lone adult counterparts. And with our prediction, we didn't be able to say, certainly that change was caused by coyote. But the one thing we could say is that coyote activity was a strong negative predictor of only fawning dial activity. And so we've still got more work to do on this. This is an unfinished project, but I thought that those preliminary results were kind of interesting. And what this potentially is saying is that when we're starting to look at how animals, especially large mammals, are adapting to urbanization and how they're going from more rural to urban landscapes, we may want to start considering how inter-Pacific differences in those responses may be affecting how those species are actually existing with those urban areas. So with that, thank you very much for listening. I've got a ton of project partners on this page. So I just wanted to kind of show them here. Sageline Collaborative really runs the citizen science or community science effort for this project, which is now up over 1,000 different volunteers that are actively going out and setting up these cameras. I was going to show a map, and I included a map of the study site in this presentation, realizing that I had said there's this giant juxtaposition between impervious cover and non-impervious cover, but I added it to the presentation on my computer and not the one on my flash drive, so it's not there. But yeah, other than that, thank you very much for listening, and I'm happy to take any questions if you have. Yeah, Kate.
Yeah, awesome talk. Do you think these results would change in the winter when coyotes often utilize snowpacks to?
Great. Well, thanks y'all for coming to my talk today. I don't think I need to convince this audience that cities are focal points of massive environmental change. Really interesting locations to study environmental change on wildlife populations, and that environmental change often can have negative consequences for biodiversity. But there's success stories and success stories that we've heard in this conference. Lots of talks on coyotes and their persistence in cities, their recovery in cities. This is a coyote walking down Michigan Avenue in the loop of downtown Chicago. Just remarkable. And I'm really interested in how are wildlife species able to persist in cities? What does that look like? Are there plastic changes in their behavior? Is there evolutionary adaptation? We've certainly found lots of case studies where we know that for species that persist in both cities and in surrounding rural areas, that they often look phenotypically different. Right, so you've got Tungera frogs in city populations that have more complex mating calls than frogs outside the city. Famous case, Anolis lizards, longer limbs, more lamellae in city populations, maybe to help climb over impervious surfaces compared to forest populations. Acorn ants, the work out of Ryan Martin, Sarah Diamond's lab in Cleveland, showing that the thermal tolerance of acorn ants for warm temperature is higher among urban populations of acorn ants, whereas those surrounding rural acorn ants are more adapted to cool conditions. So lots of examples of phenotypic trait variation between urban and rural organisms within species, and less is known about the mechanisms generating that trait variation. And so that's what I'm really interested in. I'm specifically going to focus on evolution as a potential driver of trait variation between urban and rural areas, so traits that have a heritable basis. So here in the bottom right, just showing what we might find in terms of trait variation between urban and rural areas. A cline, I'm going to show U, an allele frequency on the y-axis. And so we just have a simple climb where U has a higher frequency in the urban area and a lower frequency in rural populations surrounding it. And we might look at a climb like this and jump to the conclusion that, wow, I mean, cities are so dramatically different than surrounding rural areas. There are all these novel selection pressures that this is maybe an indicator of urban adaptation. No doubt that there are novel resources and conditions, the urban heat island effect, more impervious cover in cities. All right, so that's one plausible hypothesis. And we might go out and collect some fitness data to look at a key postulate of natural selection, differential fitness between genotypes in different environments. And we might find a pattern like this, right? Where in the urban population, the U genotype has a higher fitness than the R genotype, but in the rural population, it's just flipped, right? The R genotype has a higher fitness than the U genotype. So selection favoring different varieties, different trait variants in urban and rural areas. But we have to remember that these urbanization gradients are also gradients of fragmentation, and that can mediate non-adaptive evolutionary forces, namely genetic drift and gene flow. So the networks of transportation, roads, rails can cause reductions in gene flow. Species that persist in cities might persist at lower population sizes where the strength of drift is high. Species can be translocated into cities, right? So there could be founder effects. All of these are non-adaptive mechanisms that might generate clines. So pairing clinal data on trait variation along urbanization gradients with fitness can be really key for differentiating between adaptive and non-adaptive mechanisms. If it's all just drift in gene flow, we should find no difference in fitness between the U and R genotypes in both urban and rural populations. So it's important to say, though, that these are not mutually exclusive mechanisms, that you can have a combination of adaptive and non-adaptive mechanisms along urbanization gradients. So one hypothesis is that maybe there is urban adaptation. The U genotype performs better than the R genotype in the city, but rurally there's no difference in the fitness between those genotypes. You've got primarily drift controlling frequencies in the rural area, but selection in the urban area. That would generate a cline like this. And the reverse would also generate a cline like this. Perhaps there's no urban adaptation, there's actually rural adaptation. The R genotype performs better than U there, but whatever selection pressures we do not use relaxed in the city, and U is able to persist there, just random luck and drift. So I'm interested in testing these kind of hypotheses, and I'm going to use eastern gray squirrels as a kind of a case study to do so with the model pigmentation system. The eastern gray squirrels are a great study organism to look at urban evolution. First of all, pretty much everybody knows squirrels. They're highly visible. They're diurnal. They have this cool coat-color polymorphism that's clearly visible. This is a polymorphism that's driven by a little deletion in the melanocortin-1 receptor gene, which underlies melanism in a lot of vertebrate species. And historically, we know that melanism in gray squirrels is more common as you move north. There's a latitudinal large-scale cline. There's been some older physiological ecology work suggesting that melanic individuals have greater potential for non-shivering thermogenesis at cold temperatures, to your question about thermoregulation. And so that might drive that latitudinal climb. But I'm interested in the smaller scale stuff, what's going on along urban to rural gradients. Anecdotal reports from European settlers making notes about the fauna and flora suggest that the melanic morph was very common in the northeastern United States, even in rural areas outside of developing cities. And anecdotally today, that doesn't seem to be the case. So there's been anecdotal reports of declines in melanism in rural forests, and they're kind of hanging on in city populations. So the first thing I want to do is, do we have a pattern, right? Are there clines in melanism along urbanization gradients? And then I wanted to dig into, well, what are the evolutionary mechanisms that might be driving that? So to test for a cline in melanism along an urbanization gradient, our focal city, although we're doing work across many cities, our focal city is Syracuse, New York, and upstate New York. And so you can see Syracuse here. We set out a bunch of camera traps at 49 sites. Those are the starred locations. And then we did point count surveys at an additional 20-some sites, 27 some sites. And we did point counts at the camera sites as well. So we have both count data and detection, non-detection data for each color morph of squirrel on a daily basis for over a year. We had the cameras out, and we did 1 to 7 point count surveys at the sites as well. And so we built an integrated hierarchical model to estimate the abundance of each color morph at each site. So we had to integrate the count and the detection, non-detection data. And we did that by combining an admixture model for abundance with a Royal-Nichols model of abundance using detection, non-detection data, where the common parameter is P, the individual probability of detection at a given site, at a given time, in a given survey. And so we modeled that detection probability as a function of temperature. Squirrel activity peaks in the fall and in the spring when temperatures are somewhat moderate. Their activity goes down, does not zero, in the winter and summer when temperatures are at their extremes. That's why we included a quadratic effect of temperature. So that was the detection process. And then for the state process for abundance, we modeled the overall abundance of all squirrels as a function of distance from city center. We use distance of city center because Syracuse has this kind of nice circular, simple pattern. And so distance from city center as a spatial index is strongly correlated with, you name it, any urban index, road density, settlement density, whatever. So we use that as a nice spatial index. And so that was our model for looking at changes in abundance along the gradient, which we expected, squirrels being an urban exploiter. And then we used a binomial function to model the probability or the proportion of squirrels at a given site that were melanic. And we also modeled that proportion as a function of distance from city center. And that's our way of inferring whether there was a decline in Syracuse. So I'll show you the data here. X-axis is distance from city center. So 0 is central Syracuse, going out to about 25 kilometers south of Syracuse. Y-axis is total abundance. So you can see a really strong decline in abundance. Squirrels are way more abundant in the urban part of this study area, and they decline as you move away. The circles represent our inferred estimates of abundance at each particular site. And then what we're really interested in is the decline in melanism. So Y-axis here is the proportion of melanic squirrels at each site. And so you can see that in the city center, Syracuse, about 50% of squirrels on average at each site are melanic, and that declines to on average under 5%, 20 to 25 kilometers outside the city. This is a pretty remarkable climb. It's a pretty strong climb. There's no doubt variation among individual sites. We're working on kind of digging into some of that intra-city variation in melanism and linking that to some other indices of urbanization. But this was the big pattern that was our first step we kind of needed to nail down. This is consistent with a previous look at declines in melanism that we published last year using iNaturalist data. So we dug into the iNaturalist data. There's over 100,000 observations of squirrels. We classified them to coat color for 43 cities that had at least 100 squirrel observations. And so you can see the latitudinal decline here. You can see that melanism with the pie charts, the black shading shows the proportion of melanic squirrels higher as you go north. But what we found is a consistent main effect of urbanization, a positive effect on the probability that an individual had the melanic morph. A lot of variation among cities, no doubt about that, in both the intercept and in the slope. But we found a main effect suggesting that where melanism is common, especially in the north, we tend to find these declines where they're more common in cities. So this seems to be, there's some evidence for parallel evolution. And so now what about the mechanisms? Is there differential fitness between morphs in each environment? My former postdoc, John Vanek, who's now with Heritage in New York State, did this experiment where he translocated 76 squirrels from Syracuse into rural areas. So we had rural translocation sites, 3 of them, and then back into urban areas as sort of a control. We translocated both morphs, and then we monitored survival of individuals for over a year with standard VHF radio telemetry. They all had mortality signals on them so that we could tell whether a mortality signal went off. Just some pictures of us attaching a collar to an individual. We anesthetize them with isoflurane so that it's safe for us and it's safe for them. Then we put them in the recovery room, a little tote here, give them an apple and some Gatorade. They do quite well, let them loose, and off they go up into the trees. We translocated 18 to 20 individuals of each morph into each environment, urban and rural. And then when we recovered them, when we found a mortality signal, we had to classify mortalities, and we did this at different levels. So there were some mortalities that we were pretty certain it was a predation event, or definitely a confirmed, mortality. And so not necessarily a predation event, but a confirmed mortality. So this would be a case where, like in the upper left, an individual was shot by a hunter. In the bottom left, you might not get a carcass, but you get the collar that has a really deep bite mark, not indicating not like chewing of a rodent, right? But a kind of deeper tooth mark from an incisor. So we had 16 confirmed and probable mortalities. There were other cases where a mortality signal went off, but we couldn't retrieve the unit. It was up in 50 feet in a canopy, or a private landowner wouldn't let us onto their site to retrieve it. Or we recovered the unit, but it had no signs of bite marks, right? So we said these are possible mortalities. They could be slipped collars, okay? So we have two kind of layers of categorizing mortalities. We use the confirmed and probable mortalities to fit a Cox proportional hazard regression, looking at how the hazard of mortality was a function of environment, morph, and the interaction between the two, the interaction being of interest here for testing for differential or divergent selection. We considered additional covariates, body mass, sex, collar size, temperature during the first week of the release. None of these are important, so we kept the model nice and simple. So I'll show you the results here. These are survivorship curves in the rural and the urban environment. And then black is the melanic survivorship curve, gray is for the gray morph. The steps down represent a confirmed or probable mortality, and X represents a censored individual, an individual that we knew survived up to that point, but then there were maybe a possible mortality. We couldn't confirm the mortality. They contribute to the analysis because we know they lived for a certain time, right? So that contributes to the survival analysis. Clear pattern here. There's an interaction effect. What we find is that melanic survival is much lower than gray survival in the rural woodlands, but in the urban areas, there's no difference in survival at all, suggesting that we have selection against the melanic morph in the rural areas, but primarily drift controlling more frequencies in the urban environment. So, the next big steps for us are to kind of dig into, well, what are the mechanisms? What are the ecological pressures or selection pressures that are driving adaptation in the rural environment? And one of the first things that come to mind and kind of thinking back to Jess's talk earlier is predation. We know that squirrels in rural environments are predator limited from GUD studies and from some work on food supplementation. So human hunters have been important causes of predation of squirrels in rural areas, but also a host of predators, right? And Jess showed us this morning that the black morph stands out, right, more than the gray morph in rural secondary forests. So perhaps they're getting eaten more. We got to confirm that with some actual data on attack rates. In the city, as predators maybe diverge, right, their feeding behavior to other things, novel food resources, maybe they're not eating squirrels as much in cities, so it doesn't maybe matter as much that squirrels are standing out, black morph is standing out. So what's causing, melanics to persist in the city? Well, maybe relaxed selection of predation. Cities are kind of a refuge for this interesting aspect of biodiversity, this color morph polymorphism. How do they get there? Well, there's all these introduction events. I mean, here in DC is the most famous. Teddy Roosevelt's administration, they introduced 18 black squirrels to the National Zoo. It was a trade deal that we struck with Canada. We sent them some gray squirrels in return. There are all sorts of cases of black squirrels being introduced to city parks, right? So they make it to some cities, selection might be relaxed, and they persist or not. It's kind of luck. But this helps us maybe explain why our corridor in upstate New York from Niagara Falls to Rochester to Syracuse to Albany, Niagara Falls has a ton of black squirrels, Rochester doesn't. Syracuse has a ton, Albany has none. Right? You get this dramatic variation, drift, kind of controlling more frequencies in the cities, maybe helps us explain why that can be. So in summary, are there urban-rural declines in melanism? Yes, with our standardized data set in Syracuse, we find a pretty strong decline in melanism. Is urbanization causing selection on coat color? I think yes. Thinking about urbanization as a broad force that affects the environment across scales, including the way it's changed our rural forests, right? As we've developed that landscape for agriculture and then released that pressure as agriculture shifted to the Midwest. I want to make the claim, the take-home point here, though, that urban evolution is not necessarily urban adaptation. Here we have an interesting case where the black morph is not necessarily adapted to the city life, right? It's actually selected against rurally, and that might help explain why we see these declines in melanism. So cities could be really important places for refuges, for relaxed selection and persistence of interesting traits. Next steps, we're looking at the genomics of adaptation with my colleague, Gisela Coccone at Yale University, looking for signatures of selection at MC1R. We're going to look at ecological causes of natural selection with some studies of attack rates on morphs in urban and rural environments. And we're really interested in some of the intra-city variation, but also among city variation. The iNaturalist data has a lot of biases. We don't get many rural observations. We actually want to revisit the parallel Klein's idea using standardized camera trap surveys. And we actually have about 30 cities that have been working with us, including through UWIN. So if y'all have camera traps in a city and you have gray squirrels in the Milana coat color and you haven't been involved with us, let me know if you want to get involved and do a few weeks of camera trap for us to contribute to that data set. So happy to take questions. Yeah.
That was really cool. Thank you. I'm interested, obviously this is all very hard to do. But right, survival is only one piece of the